So we've gone and collected our water, and now we're going to set up for our primary production experiment. So I have here our dissolved oxygen sensor. Here's the probe for it. And we've got an experiment where we're going to test low light, which is going to be covered in these mesh screens. We've got a light back here that's on a timer. And then we have our highlight bottles, which are regular glass BOD bottles. And then we've got our dark treatment, which are bottles wrapped in foil so that no light can get to these bottles. So we've collected our water in these amber containers, and we're going to go ahead and start filling up our bottles. So I'm going to mix our sample first. I'll begin with a highlight bottle. And we're going to gently pour our sample into our experiment bottle. And now we want to take our initial oxygen measurement. So we remove our probe from its storage bottle and insert it into our sample. So it's going to overflow, but that's good because we don't want any bubbles inside of our sample. Bubbles introduce oxygen. So all the oxygen that we read in our sample, we want that to come from our phytoplankton that's in our water sample. And we wait for our oxygen to stabilize. All right, so this sample has stabilized to 7.42 milligrams per liter of oxygen, and the time is 9:52 a.m. So we're going to repeat this procedure for the rest of our bottles. We're going to record the initial dissolved oxygen, record the time, and we'll come back in 24 hours and see how much our phytoplankton have produced. All right, so it's been 24 hours. We've let our experiment run overnight, and we're gonna start taking our dissolved oxygen measurements, our final dissolved oxygen measurements. So I'll place this here so you can see. Remember that we left these without any bubbles, so we can see that there are some bubbles here at the top of our jar. So that means all that phytoplankton, they've been photosynthesizing and releasing oxygen into this bottle. So it should be super saturated because we can see these bubbles. So we should get some nice large numbers. So we'll start with our high treatment 1, and I'll ready the probe. Get a good seal, and we'll see what we get. All right, so our final dissolved oxygen concentration in our highlight treatment is 10.14 milligrams of oxygen per liter. So that is definitely an increase yesterday. So I'm gonna go ahead and record all of our final dissolved oxygen for all of our treatments. So for our dark bottle, we can notice that our oxygen has gone down in the last 24 hours. And this is because of respiration. So there are bacteria in there that are taking up the oxygen that's in the bottle and respiring. Our phytoplankton are also respiring. So at night, they use oxygen to do cell repair and certain things within the cells. So they don't always release oxygen as a byproduct. Sometimes they have to use it. So this is why we do a dark bottle experiment, so that we can measure how much oxygen is used during the process of respiration. So now we'll move on and keep going, measuring our samples. So now we'll take a look at the data and do some number crunching.